Okay. Now I think Nadine can unmute and start the presentation.
Okay, hello everyone. We are here as group number 21 from Rwanda with the title Enhancing PETase Thermostability Using Nus-A Protein. The presenters are me, Nsengiyumva, Ishimwe, Nadine, and my colleagues.
Ntwali Salomon.
Nestor, Irakarama.
Harerimana Jean-Paul.
Musana, Habimana, Arsene.
Yeah, and we are presenting this project to the intermediate course in Bio for all Africa 2024. Next slide, please. Yeah, so plastics are global protein due to their inability to be biodegraded, with the global production of over 350 million tons per year in different sectors such as industrial machinery, packaging, building and construction, and other places as they are shown in this slide. And all these plastics that are produced, the waste that are produced from these plastics is over 70% of all the plastics produced. And these plastics can be treated, this plastic waste can be treated by either incineration, they can also be recycled, and most of them, which is about 40%, is landfilled, and others are lost in the unmanaged dumps or leaks. PET is among the most common of these plastics, with over 56 million tons produced annually. And the green economy ensures a complete cycle where the products that are produced can also in turn be reused into waste, and then the waste can be used to form other products. Now let's look at how we can create the plastic circular economy. Next slide, please. So currently the disposal options that are used, there is the energy recovery or incineration, where the wastes are burned to generate energy, and then this energy is used to power the industries. There is also downgorging and reuse, where they reduce the thickness of the products, the plastic products, and then reuse them. This can reduce if this can be done, can reduce the waste up to 73.6%. There's also landfilling, where the plastics are disposed in landfills. But since they are not biodegradable, they can persist in the environment, causing problems. There's also bio-based methods where they can use enzymes like PETase to degrade the plastics, the PET-based plastics, but they are less efficient due to the thermostability problem of the PET. Let me call my colleague to continue to the problem statement, telling us exactly what the problem we are trying to solve. Thank you.
So thank you so much, Nadine. I'm here by the names of Harerimana Jean-Paul, and we are going to see together the problem statement of that I'll consider during the study of our topic. I've seen that currently methods like incineration and landfill that are used in the management of plastics are not eco-friendly. Next, the bio-based methods that are used like enzymatic degradation are less efficient and often requires the significant research and development studies. After seeing that, we have come up with our proposed solution, where we are required to enhance the PETase activity through improve the thermostability of the PETase enzyme. The PETase plastics requires the higher temperature to expose the crystalline regions that are resistant to the PETase. These crystalline regions are taken with the covalent bonds that are made during the vulcanization of such plastics to make them thermotolerant. The high temperature is think to weaken the intermolecular forces due to the higher molecular weight. Therefore, as a group, we decided to improve the thermostability of PETase, allowing its activity to higher temperature. From the previous studies, we have seen here in this graph where they compared the wild type and genetically modified enzymes. Normally, they take 1 milligram of the enzymes and treat the PET plastic of a gram, and then they analyze the rate of degradation in order to produce the aromatic compound. So as a simple analysis, you can see the wild type and the genetically modified, all of them are treated on temperature of 30 and 40 respectively, where the wild type showed them that they are most efficient on temperature, the optimum temperature of 30, and upon reaching increasing the temperature to 40, they are denatured and the rate of degradation of PET to aromatic compounds decreased. While for the genetically modified enzymes, the efficiency of degradation was increased upon increasing the temperature to 40 Celsius degree. So we are proving that after making our modification to the normal PETase, our enzymes will be able to work on a temperature which is mostly can reach the temperature above 40 Celsius degree. So I may invite my colleague to continue to see how this modification will be done. Thank you.
Thank you, Jean-Paul. And as our aim of this project is to produce the PETase with enhanced thermostability, we will first do a expression vector construction, and the expression vector construction will be done firstly by synthesis of isolated IS-PETase mutant gene from the wild type IS-PETase that was firstly isolated from Idonera sakaiensis bacteria from Japan. Secondly, after synthesizing the mutant gene, we'll clone this gene into a PBAD vector for gene amplification, and the amplification will be done using PCR. The fourth method will be the isolation of Nus-A gene construct from E. coli. Nus-A gene encodes for a protein Nus-A, which have been found to process chaperone activity as well as thermostability activity when bound to some other protein domains. And then we will assemble the Nus-A construct with the IS-PETase mutant gene on the plasmid PET30A, which is commercially available, and you can just find it on the Agent company, to form the recombinant construct, and the assembly will be done using Gibson assembly. After making the recombinant construct, we will transform this into E. coli as a chassis. E. coli is chosen because of its two important properties. First, easily genetically manipulated, and second, also grow at very cheap substrates. Then we continue with the protein expression and purification. After making our recombinant construct and transforming it into our chassis E. coli, we will culture the E. coli cells to obtain high amount of culture using Luria Bluff at 37 degrees Celsius to obtain the optical density, which is necessary for protein expression. We'll do protein expression using the inducer IPTG, and we culture this we incubate this culture at 20, 25, and 30 degrees Celsius for the optimal condition of our expression vector and our expression bacteria. Then we will harvest after expression and growth of our inoculum. We will harvest the cells using sonication method. The sonication method uses ultrasound waves to heat the cell and lyse them, and the protein will be released in the buffer A that we have produced, and this buffer will maintain the acidic condition. And then the supernatant, which will be formed, the buffer will contain our protein of interest, will be treated with the high strap column, and this high strap column will be mixed with imidazole to remove the non-specifically bound proteins and only remain with the proteins with Nus-A PETase complex. And then we'll purify or concentrate our protein Nus-A PETase complex using Bradford reagent, and the proteins the protein concentration will be measured spectroscopically. Thank you. I may invite my colleague to continue with the next steps.
Thank you very much, Nestor. After successful extraction and purification of our fusion protein, we'll proceed with the characterization, whereby we'll conduct thermostability measurements and biodegradation activity. In these measurements, we'll be comparing the wild type and the mutant, and for the thermostability test, we will conduct it using the fluorescent method, whereby we'll use a dye, the protein thermoshift dye, to measure the stability of the protein at different temperatures. The temperatures will vary using the RT-PCR machine at a rate of 0.5 degrees per minute from 25 degrees Celsius to 100 degrees Celsius. In order to monitor the biodegradation of PET, we'll use two different types of PET plastics, the PET-NP with low crystallinity and another PET for high crystallinity, to also measure the effect of the degree of crystallinity in PET to PETase activity. And in order to measure this effectiveness, we'll be considering three aromatic products of this process, which are TPA, MHT, B8, and then we'll use high-performance secret chromatography to quantify the amount of the aromatic compound produced. So after successful implementation of our project, we looked at different competing solutions, and there are very many, since this is a field of this is a very hot topic in research. Other research went over engineering microbes, engineering enzyme specific, and also even engineering the metabolic pathways as a whole to create valuable products. One particular research went over protein scaffolding of self and non-self in order to improve the thermostability of PETase. They used the SpyCatcher technology and GFP nanobody fusion protein. For the SpyCatcher, it displayed significant increase in thermostability, but a reduction in efficiency, as there might have been hindrance in the folding of the protein. For the GFP nanobody, it displayed both increased thermostability and also increased efficiency. This proves that fusion proteins are much more efficient in this process, giving us also hope that ours might be competitive in this area. I can invite Arsene to proceed with the conclusion.
Thank you, my colleague. So as the conclusion, as my colleagues say, the PET plastics play a crucial role in our everyday lives, from packaging to manufacturing, and this is because by their durability and versatility. So, however, this issue of durability of this PET cause or lead to the severe environmental pollution, as this plastics does not decompose very easily in nature. So they have discussed that the traditional methods of disposal, such as incineration and landfilling, may cause the significant environmental degradation, such as release of harmful pollutants into the atmosphere, and while landfilling takes up the very valuable space and poses long-term ecological risks. So in response to these challenges, the scientific community have intensively researching bio-based solutions. So our proposed approach focuses on enhancing the enzymatic degradation of PET plastics using the enzyme PETase. So, and this enzyme have shown previously the promise into the breaking previously have shown the promise in the process of breaking down of the PET plastics into the small monomers. So to address this, we are expressing the incorporation or the using the NUSA protein with PETase, and we aim to develop the modified PETase enzyme which retain its higher activity at higher temperatures, and those temperatures which are closer to those which are into industrial settings. So this enhancement will potentially enable more efficient and effective degradation of PET plastics, and this will make process not only eco-friendly but also economically viable. So our innovative approach to improve the PETase with the incorporation of NUSA protein represents a significant step towards sustainable plastic waste management. This bio-based solution has also the potential to transform how we handle PET plastics, and it will offer more efficient degradation method that aligns with the industrial conditions. And by advancing this research, we will contribute to mitigating the environmental impact of plastic pollution, and this will also pave the way to the cleaner and more sustainable future uh, against the plastic pollution on the earth. Thank you for your attention and interest in this vital area of research. We look forward to any question or discussion or how we can collaboratively advancing our research into product-based